Well, hello, everybody. Thanks for coming. Did everybody have a good day today in Ukraine? Excellent, excellent. We did too. Thanks so much. For those of you who haven't met, my name is Mohammed Noor. I'm a professor at Duke University, and the talks I've given so far have been about evolution. This one is a little bit of an outlier. The focus of today's talk is, well, you can see from the title, is genetics, genomics, and you don't fear your genotype. And I'll be focusing especially on this last part. Many of you are aware that there's a lot of direct-to-consumer genetic tests out there now. And ultimately, what I'm leading up to through this entire talk is what is the efficacy of those things and what are things you need to look out for if you're considering doing one of those direct-to-consumer genetic tests. Now, one thing I will say right up front is please let me know if I'm speaking too quickly. I do get excited sometimes, but please let me know when I'm actually speaking too quickly so that I can try to slow down. Oh, you're right, it did not. Okay. Julie's keeping an eye on me. Thank you. So the other thing I'll talk about is, or the other thing I'll mention briefly is that tomorrow I'll be doing a lab. And in that lab, we'll be isolating or precipitating DNA from cheek cells. So what you'll do is you'll essentially be swishing some Gatorade around in your mouth. You'll spit it out into a little tube, and we'll actually pull the DNA out of that. Now, one thing I want to emphasize, this is not a genetic test of any sort. There will not be any conclusion from this about disease predispositions or ancestry or anything like that. It's literally just to see your DNA. But in order for it to work, please avoid coffee beforehand, which I know that might limit the attendance a little bit. But brushing teeth is fine. I mean, what will be happening is they'll be getting it out of your cheek cells itself. The problem is the coffee is going to take out a lot of stuff from your cheek cells. I guess if you drink coffee, try it. Slug it really fast, so it's not going around your mouth very much. I know. The longer, the better. Basically, if you don't have it, I guess it depends on which one you do. So I'm doing one set in the morning and one set in the afternoon. For the morning set, probably best to just avoid it the whole morning. For the afternoon set, if you have it at 8 a.m., probably it'll be okay by 1:30 p.m. It's a little bit of a challenge. It'll still work. If you do it with coffee, it'll still work, but it just won't work nearly as well. So the amount of DNA you'll get is a lot less. Tea's fine. No, specifically black coffee or coffee with coffee that at one point in time was black. So if you mix it with milk, it's still just as bad. Basically, yes. It's just going to make it much harder to get them out into the Gatorade. So, sorry about that. The morning and the afternoon are exactly the same thing. That's a very good question. The morning and the afternoon are exactly the same thing. So if you're like, I've got to have my coffee at 8 a.m., then you can have it at 8 a.m. and then come to the afternoon session, and that should be fine. Yes, do not go to both. Otherwise, you'll just have 2 tubes of your DNA. It'll be exactly the same thing in the morning as in the afternoon session. Yeah. Any other questions on the lab? Yes. The total number we can do is 150, so I think we'll be okay. What was your question? Sorry. It's in your booklet. I don't remember exactly, but I think it's 10:30 and 1 or something like that, or 10 and 1. Oh, there we go, 10:30 and 1:30. No, it'll basically just be this. I mean, we'll talk a little bit informally, but it'll basically be just that. So you can think of it as the lab associated with this lecture, sort of. Okay? All right, let's get started on the meat of today's lecture. So many of you know there's been a lot of disease genes in the news. These are some sample headlines I just pulled off the web. So things like Parkinson's disease gene is found, gene increases diabetes risk, gene increases Alzheimer's risk, big batch of breast cancer genes. These are headlines you see all the time in the media. And the media is, of course, one of the worst places to get any sort of scientific news, but nonetheless, that's out there. There's all sorts of other genes that show up, too, things like researchers identify the alcoholism gene. Oh. It's not Dr. Krause. They shouldn't come on.
Very good afternoon, ladies and gentlemen, and welcome back on board the Elegant Rotterdam. Captain, speaking to you from the bridge, with an nautical update. Currently, the final passengers to return from the shore are boarding the vessel, and as soon as they are all on board, we're taking our gangway and get on the way. We'll slowly sail, backing the vessel out of the harbor, and once we're well clear of the breakwaters, we swing the vessel around to proceed to the south. Then they will cross the Black Sea, a direct course to the Bosporus, ETA 10:30 tomorrow, on a southwesterly course. Weather forecast was a bit bumpy last night. should be fine tonight with less swell and less wind, so we should have a smooth ride all the way to the Bosporus. I hope you enjoyed Yalta, a very interesting place. Nice microclimate they have here, so a lot of vineyards and so on. A lot of things to see. I hope you enjoyed it. They were surely grateful to have us here today. And I hope you all enjoyed. Have a wonderful evening on board the Elegant Rotterdam. And now hear a word from Tom, your cruise director, with an update of the activities and tonight's entertainment. Tom.
Thank you very much, Captain. And good evening, ladies and gentlemen. Welcome back. For those of you that went ashore today, a couple of things to remind you about. First of all, is we need to have your passports back. We'll most likely give them back to you later, but we'll need them to clear the ship when we get back to Turkey and back into Greece. And I'll tell you more about that at a little talk I'm going to have tomorrow. Tonight, you can look forward to a margarita sail away with Janice and the Hellcats happening around the Lido Pool midships. It starts at 4:30. That's now. And then also, your shops on board are having another one of those fun sales. Today it's a Slavic Bazaar, so that goes from 6 until 10 this evening around the atrium on deck number 5. And you might want to ask your spa manager about what a 20-20-20 special is. Looks like something special to do with many different treatments they're offering, so go to the Greenhouse Spa on deck 8 to find out all about it. Wine in under 30 minutes with Cellar Master, the amazing Zoltan. There's a wine cellar. That's on deck number 4, I believe. It's 5. Pub trivia, 6 o'clock in the mix with your friendly DJ Renee, something we like to do almost every night. Grab a drink and learn some trivial answers with your DJ Renee in the mix. Pub trivia. Tonight is also returning our big monster karaoke machine, 40,000. There's 4,000 songs in that machine, and you can pick one of those. Last time it was pretty popular, so we're bringing it back again. We're starting at 9 o'clock in the Crow's Nest with our karaoke, and we'll go until you're tired of singing the songs. And for our showtime tonight, it's Dinner Bells. It's the stars of the showroom at sea with a fun show that takes you to the music of the 50s and 60s. So join myself and the stars of the showroom at sea, either at 8 o'clock or at 10 o'clock. You just pick the time that works best for you. And I'll mention right now that our scenic sailing will begin at 11 o'clock, actually, in the morning. We're going to be picking up the pilot by 1/2 or 1/4 till 11. And then at 11 o'clock, we'll have scenic sailing. Jai will provide a little commentary as we begin our way down back to Istanbul. But at 10 o'clock in the morning, join me in the showroom at sea as I provide a short, informative, and entertaining talk about your transition from ship to shore when we get to Athens. I know it seems a little early, but it's our last opportunity to do this. So join me at 10 o'clock in the showroom at sea for your disembarkation and crew farewell event. Put together a little video, show you some of the crew around the ship. I hope you'll join me. But for whatever you do right now, remember that every day's a gift. Wherever you go, do it with a smile. I'll see you around the ship.
There we go. Got the gift and the smile both. So as I was saying, several other genes have come up in the news as well, things like the alcoholism gene, the so-called fat gene, I love that one, the gene for left-handedness, the gay gene, things like that. These headlines come up all the time. And again, these are very poorly worded, and we'll talk about why in just a second. Now, one of the questions that when I talk to the general public, you guys probably would never ask this question, but when you talk to the general public, one of the things they always ask me is, so what's the status with that raging debate between nature and nurture? Where do you stand? Like, you know, there's people who just say, nature, or something like that. That's never the case. By the way, for those of you who can't read this comic, it says, well, you walk like a duck, you quack like a duck. May I ask who brought you up? So the other issue is that some of these disease genes don't always hold up. So scientists debunk the so-called fat gene. I love this headline. Exercise blocks the effect of fat gene. Wouldn't have thought of that one ever. Breast cancer gene risk may be overstated. Now, related to this and more central to the topic of today's talk is this idea of the direct-to-consumer genetic tests. There's been a lot of controversy associated with them. So, for example, in July 2010, the FDA wrote a very strong letter to several of the makers of these companies, including 23andMe. That's just one of the ones we'll talk about the most today. But they faulted these companies for not having these genetic tests approved in any sort of way ahead of time or seeking some sort of government approval. Walgreens was initially going to market one of these personal genomic tests, and they actually backed down at the last minute, deciding not to do it. The company that they were going to order them from still does provide them, so people can still get them, but they just don't have them on the shelves. Nonetheless, and this is actually very recent, nonetheless, this is something that a lot of people are very interested in. This is from the October 5th issue of Nature magazine, which if you're like me and have totally lost track of days, that was Istanbul day 1. But as you can see on this, about half of the respondents to a poll from Nature magazine said they would take a direct-to-consumer genetic test if given the opportunity. Another almost 20% have actually had one done already. So adding those up, that's over 70%. Now, this is a very biased sample because these are readers of Nature magazine rather than just anybody off the street. But there's only a very small fraction who said they have not and would not take such a test. Only 15% of people said that. Now, if you're curious about those people who did actually these tests, they had a little bit about what was motivating them. And they divided them primarily into biology, medicine, and other. Curiosity was the biggest factor driving people's interest in this. But especially among the more medically oriented, specific health risks and general genetic influence on health were big motivating factors. That is actually a bit of a shame because for most of these direct-to-consumer tests, that is actually not a particularly good reason. And we'll talk about why that's the case. If you have a specific risk factor, it could be. But if you don't have an a priori reason, that's not necessarily a good way to do it. In terms of the types of tests they did, the vast majority did the single nucleotide polymorphism test, which I'll talk about. And a lot of them used the company 23andMe in particular, which I'll show you some sample data from a little bit later on. So let me start by giving you an outline of my talk for today. The first part will be very basic because I know people are coming from diverse backgrounds here. We'll talk about genetic inheritance and just how it works. We'll talk a little bit about the genetics of human diseases, particularly those diseases that have a genetic component, of course. I'll just touch very briefly on this nature versus nurture idea. And then we'll go into sort of the meat of the talk, these ones down here. The success of identifying genetic bases to common human diseases and the benefits and risks of actually doing these direct-to-consumer genetic tests. Okay? Any questions so far? Feel free to interrupt me if there's questions. And again, please let me know if I'm speaking too quickly. Yes? That's a different sort of test. That's not the same kind that I'll be talking about. That's a different sort of test. We can come back to that in the question session if you like. All right. So the basics of genetic inheritance have been known long before anybody had any idea of what the genetic material was. So here's a lot of examples I just pulled off the web of familial resemblance. But many of you know this as well. Most people tend to look more like their parents than like somebody picked at random from the human population. And these are just some examples. If you're curious about my family, there's my parents over on the left, and there's my kids there. You can see he has my enthusiasm. But again, we all look more similar to each other than we do to people chosen at random. You've seen my wife too right there. She's not in this particular picture because she took it. But they don't look as much like her, that's true. She is the mom, though, I promise. Now, Darwin attributed this family resemblance to what he called gemmules. Now, he was very unfamiliar with what was actually causing this resemblance. This was a problem for him because you may remember from my lectures before that in order for natural selection to work, you need a mechanism for heredity. If you don't understand how heredity is working, it's actually difficult to understand how natural selection is operating. So as he wrote in this letter to Galton, he said, if I ask myself how you derive and where you place the innumerable gemmules contained within the spermatozoa formed by a male animal during its whole life, I cannot answer myself. Typical Darwinian kind of backwards writing, but it makes the point nonetheless. Now, at this point, Mendel's pea experiments had been done, but Darwin was unfamiliar with them. So let's talk a little bit about those. But first, let me show you one of my high school errors. This just illustrates some of the issues. So in 10th grade biology, my biology teacher's name was Mr. Bennett. He asked, if you were to cut someone's left arm off and they have kids, would the kid have 1 or 2 arms? That one was easy. That one was easy. I got that one right. But he also asked if you were to cut someone's left arm off and cut their kid's left arm off, repeating for 20 generations, would the 21st generation kid have 1 or 2 arms? And there were my answers. This was before I decided to be a biologist. Now, the reason for this is very important. The reason for this is because we have particulate inheritance. We have particulate inheritance. We are not inheriting something based on the experience of our parents most of the time, but we're instead getting these particles, which are actually giving some of the genetic material from the parents to us, from us to our kids. So in this regard, we're not a simple copy of our parents. We're not necessarily the midpoint of our parents. And you saw that from my kids. They don't look exactly like the midpoint between Julie and me. They're somewhere more to one side or the other. You can have kids which don't match either parent, such as having blue-eyed offspring from brown-eyed parents. So this is a case where traits are being masked. This comes to the concepts of dominance and recessivity, which I'll go over in just a second. Genetic diseases do pop up, and all this was known long before we knew what the genetic material was. And this comes from the genius of Gregor Mendel from his pea experiments. So let's talk about those very briefly. He studied inheritance in peas, and he had a variety of characters that he was looking at. So this just shows you a subset of them. He had, for each one there was 2 types. There were round peas or wrinkled peas, yellow or green, etc. I won't go through each one, but you see the types of traits he was looking at with the peas. Now, in looking at these, he formulated some very simple principles. This is just a quick aside. You know, he's very famous nonetheless. If you can't read it, it says, "Brother Mendel, we grow tired of peas." And this was a picture from Google on Mendel's birthday. So again, he's very widely recognized there. It's great when Google does these things. But he identified these simple rules of inheritance. So he had, for example, his true breeding green pods, which I'll symbolize with big Y, big Y. So everybody has 2 copies of every one of their genes, 1 copy from their mother, 1 copy from their father. And you give 1 gamete to your offspring. So in this case, this one would pass on a gamete, which would be big Y. You can have true breeding yellow pods, which we'll call little y, and the gametes would be little y, of course, one of the 2. And then you have what are referred to as the F1. This is the offspring from a cross. In this case, when he did the experiment, all of them had green pods, just like one of the parents. And this would be their type, big Y, little y. And in this case, you have the masking of the yellow color by the green color, right? Because this has both the yellow copy variant and it has the green copy variant. But the yellow copy variant is completely masked. This is referred to as dominance. So you say that the green form is dominant, the yellow form is recessive. Okay? Many of you know this very well, but I'm trying to catch everybody up because it'll be relevant for the next part. Any questions on this? Good. Now, this one, as I pointed out, can give either the big Y gamete or the little y gamete to its offspring. And this is what he tried doing. So what happens when you breed these F1s? So these are called heterozygous. You can see where the name comes from. Hetero being multiple forms, zygous being referring to like zygote, the offspring, since it has both alleles. And you can draw it out here. Where here's the mom, has the big Y and little y. Here's the dad, big Y and little y. So if you look at what happens, half the time you give the big Y, half the time you give the little y. So if you have, in this case, the pollen from the big Y and the seed from the big Y, you get big Y, which would still be green. These both would be big Y, little y, which would also be green because they get 1 copy of the yellow, one copy of the green. And remember, the yellow is always masked. It's recessive. But if you get both of the little y ones, you actually do turn out yellow. So this is a case like what I was describing, where both parents are green, but the offspring, at least some subset of the offspring, are actually yellow. If you want to see what his actual numbers were, these are the numbers. Allele is just a form of a gene. It's very... Exactly. The capital Y or the small y would be the allele. Good question. These are actually Mendel's numbers from his experiments, 428 green to 152 yellow. And you see that fits the 3-to-1 ratio almost perfectly. Actually, some people erroneously, I should say, argue that Mendel fudged his experiments to make the numbers better. I can tell you for a fact that is, well, not for a fact. As far as we know, that is not true. That is an unfair allegation that some people have leveled at him. Okay, any questions? Good. Now, this looks pretty straightforward up to here, but these rules don't seem to hold consistently. Well, first, there are cases of intermediates. So you can have, for example, red flowers and white flowers yielding pink flowers. This is okay with respect to Mendel's rules, because you could imagine something like this, big R, little r. In this case, you have big R, little r for all these, and maybe you don't have a complete masking. You don't have complete dominance of one form. That part seems okay, but more critically is this idea of continuously varying traits. I don't know if you can see this picture very well, but these are actually people lined up by height from a university graduation. And each little notch there is everybody's height. So these people are 5'0". The white ones are female. The black ones are male. Goes up to, I think, 6'0" is about right there. These are 6'5", etc. But you see, this height is an example of something that's very highly genetic, and it's a continuously varying trait. There was a lot of debate shortly after Mendel, basically between Mendel's time and about the 1930s, about whether what Mendel saw was a fluke or whether that really was the mode of inheritance, because people would refer to this continuous variation and say, there's no way this can be caused by single genes. Now, the resolution came out in the early 1900s. What they said is there are actually lots of genes contributing to variation for each trait. So something like height, instead of being controlled by one gene, is actually controlled by, for example, 10 or 100 genes. And each gene is being inherited in the manner that Mendel described. And this can explain continuous distributions. I'll show you why in just a second. And you can also suggest that maybe these are not all dominant. As I pointed out, you can have intermediacy between two forms. So let me give you a fictitious example, but just to illustrate how this works. This is completely fictional. Let's imagine that you have 6 genes that control height. So there's in the rows there are the genes, and the columns are different individuals. So we have 7 different individuals and their genotype for these 6 genes. Now we can just say that height in inches is determined as 5'0" plus the number of capital letter copies you have across all the different genes. Right? Sure.
These are 6 different genes that, in different ways, would affect height.
These are, in this particular example, they're all affecting height the same way.
The same way.
Yeah, exactly. So we're assuming the capital letter alleles at any of those genes make you taller. The lowercase alleles make you smaller. It's a very simplified example just to illustrate the point. So in this case, if you were to apply this principle, you would count in, say, person number 1 here and see how many capital letter copies are there. 1, 2, 3, 4, 5, 6, 7. So this individual would be 5'7", right? The next one would be a little bit shorter, 5'2", etc. And what happens when you apply this? There's the numbers filled in. When you apply this, you get this sort of continuous distribution with a center around the average there. So in this case, the average would be 5'6". There'd be very few individuals that were 5'0". There'd be very few individuals that were 6'0". So this is the means by which you can actually get a continuous distribution from individual genes, each inherited in a Mendelian fashion. Yes? That would be dominance in that case. So in that case, you'd have a case where both parents are big A, little a, and the kid might be little a, little a. And that is a case where it is showing dominance. Good question. There are also, and now these are very simplified examples. It could also be something nutritional, for example, or things like that. It doesn't have to be purely genetic, but the examples I'm giving you are genetic ones. Yes? Yeah.
Slide 2 back when you got 2 heterogeneous genes.
Mm-hmm.
Is there any real reason to expect that it should come out to be 1/4 for each of the 4 resultants?
It would be 1/4 within error. So let's say, for example, you have 1 kid. Obviously, can't have 1/4 of a kid be that. But you shouldn't get, for example, 10 kids all who have the type that would be 1/4, because that would be very unlikely. Probably that would be 1/4 to the 10th power.
Well, I guess what I meant was, is there a fundamental mechanism that makes it so that that is really a fair throw of the die?
It really does most of the time. It really does, because what's happening is you're for most genes you're equally likely to give one gamete or the other gamete. And if you're heterozygous, then you're going to give either your big A or your little a. And it really is pretty close to 50-50 with some very few exceptions on that.
It depends on how the cell divides to produce the gamete. And the division is equal 50-50, so your probability is equal 50-50.
There you go. Other questions? Okay, these are great. These are good questions to start off with. Sure. Mm-hmm. It is not. I mean, people describe it as being inherited as a single gene thing. I'll come to that exact example in just a minute. But people describe it as being a simple case. It's not that simple. It's not just brown is dominant over blue, one gene kind of thing. It's not like that at all. But we'll come back to that in just a second. Now, these genes are inherited on 23 pairs of threads, or as we call them, threads called chromosomes. Now, these are long. They typically have many genes, on hundreds, or in some cases even thousands of genes. The one odd pair, which many of you are familiar with, are the X and Y chromosome. That in humans and in primates and most mammals, if you're XX, you're a female. If you're XY, you're male. And essentially, your dad is determining your sex, because your dad is the only one who can give an X or a Y. Mom is always just giving the X. So this is kind of interesting in cases of kings who were very mad at their wives for failing to give them a son. It's not the mom's fault. It comes strictly from the dad. So if dad's always giving an X, mom can't do anything about that. So that's an important facet to this. Now, these genes themselves are made of building blocks. They're called nucleotides. They're abbreviated T, A, G, and C. Those are for thymine, adenine, guanine, and cytosine. But don't worry about that. Several people ask me informally, like, how many average building blocks or nucleotides are there for each gene? The average number of coding ones for humans is about 3,000. It's about 3,000 on average. And again, these, variation at these building blocks is what causes differences in appearance, such as with the peas, the yellow versus green peas. They're caused by, for example, a C at position 5 rather than an A. That's the difference between, say, the big A and little a that I was more crudely describing. Okay. Any questions on that? I know for some of you that you've seen this 1,000 times. Now, I want to start off with a little activity. Let's pass this. We're going to pass some things around there. There are very few traits that are caused solely by one gene, or at least inherited such they seem to be caused solely by one gene. One of them is PTC taste. This is the ability to taste phenylketonuria. So we'll actually try it right now, where you guys can actually taste it right now and see whether you're a taster or a non-taster. What's that? Oh, no, it's not actually. This is chemically synthesized. It's not. Didn't come from down the hall. So we'll pass these around right now for you guys to actually test it. Actually, well, I'll tell you what. Go ahead and grab these when you get them right now, and what I'll do is I'll show you some other traits in the meantime. That way we're not holding up the talk.
It's just one each. The blue isn't any different than the white, is it?
No, the blue, white is a control. Yeah, exactly. The white is a control, and the blue is the taste one. So tongue roll. How many people here can roll their tongues? A lot of you can. I actually cannot, so I actually have to have my hand down. So this is one that, again, it seems to be inherited, like a single gene trait, but the inheritance of this one is suspect. And there's also some crazy examples out there. Like, look at that. There's some crazy examples like that. Now, in that case, the ability to roll your tongue is thought to be dominant. So basically, so let me ask you guys a question. If the ability to roll your tongue is dominant, right? Let's say you have two parents that cannot roll. Should the kid ever be able to roll? Yes or no? So remember the ability to roll your tongue, maybe. That's the safe answer. The ability to roll your tongue is dominant. Yeah, and it's one, assuming it is one gene, which in this case that's suspect. So there's several people who said no and several people who said yes. So let's draw this out for a second. Oh, I have to exit this to do that. 1 second. So we said, oops, let's see. So big R is the ability to roll. Okay. Woo, getting a little bouncy here. So big R is your ability to roll. Now let's say you have parents which cannot roll. What is their genotype? Little r, little r. Is it possible to have a kid who can roll? So in that case, it's not. The opposite is true, right? You could do the same thing. You could say if both parents can roll, this would be a case where both parents can roll. Then you can possibly get a kid who cannot. Or mutation. Well, we're taking out the idea of a new mutation. That's true. So I just wanted to show that as an illustration. Let's come back to there we go. Did it show up? So that one's kind of suspect. This one's a pretty good one. Free versus attached earlobes. So see, I've got dangly ones here too, like this person on top. Free versus attached. Yep, you're a dangly one like me. So in this case, let's see. The hanging free, the dangly ones, is thought to be the dominant trait. The attached ones is thought to be recessive. So I know there's a couple of parent-offspring pairs here. You can check and see if you have the same trait. Yes? Oh.
But about 4 weeks, they begin to flap with their tongue flat, with the exception of one known breed of China, which, as an adult, drinks water with their tongue rolled up in the poop.
I didn't know about that. That's great. That's a really cool example. Yep. What's that? Yeah, there you go. That's pull it up. Show you a couple of other examples. Class for hands here. Do this. Watch this. Which thumb is on top? How many people have right? Yeah, see, a lot of you have left. Right is recessive. Left is dominant. Isn't that weird? That's the weirdest one, I always think. I actually have, this is one of the few things I have the recessive trait for. Julie's always very mad at me because I have the dominant traits for almost everything. If you do it the other way, it feels very unnatural, doesn't it? Try it the other way. It feels really odd. Hitchhiker's thumb. See, look at this. Mine goes way back. I've got the serious hitchhiker's thumb. You have one of each, really? Maybe you're heterozygous. No, for hitchhiker's thumb, the straight thumb is thought to be dominant, and the bent thumb is thought to be recessive. Okay. Widow's peak is another one. I've definitely got the widow's peak action going on there. That's the pointiness there in your hairline. There you go. The widow's peak, this one is dominant. Oops. Oh, sorry. The widow's peak one is dominant. Sorry, I forgot to turn on the pointer thing. There we go. This one's dominant. That one's dominant. And the thumb, which one? The hitchhiker's thumb? The straight thumb is thought to be dominant. Going back like mine is recessive. Okay. Dimpled chin. Having the little cleft in there is dominant. You can't see in mine, but if you have the little dimple in there, that is thought to be the dominant trait. Absence, if you have just sort of a flat chin, that's thought to be recessive. I actually don't remember. I have to go back and look at a picture. Yes. Sure. Oh, really? Oh, there it was, hiding in there. Exactly. And that one's dominant, so there you go. And the last one I'll show you guys, well, I'll come back to the PTC because I know it's still coming around. Last one's mid-digital hair. I've got hair everywhere, but here's mid-digital hair. So when you have hair on that second digit there, that's thought to be dominant. It's harder to see on women. Yeah, it's harder to see. It happens with women too, but it's harder to see usually. It's usually like a hair or two and it's really thin. I even have that on my toes. And mid-digital hair. Mid-digital hair, having it is dominant. If you not having it is recessive. Yeah?
If these are dominant, why after so many generations of the human race doesn't everybody have them?
Ah, that's a very good question. So that's a purely evolutionary question rather than a genetic one, but nonetheless, it's a good one to ask. So this actually comes back to the principle introduced by Hardy and Weinberg. These are, I guess one was a physiologist and the other one was an English mathematician. I think Hardy was an English mathematician. It seems like the dominant one should take over, but unless there's actually some advantage to it, the abundance of that type isn't actually changing. So you have a set number, for example, or a set fraction of the population. Say 30% of alleles out there are the dominant type. There's no way for that 30%, unless they're actually spreading, to take over and make it so that everybody has it. Yeah, aside from that, as I said, yeah. Yeah, so dominance itself doesn't necessarily make it spread. It seems like it should, and that was actually one of the common thoughts around the early 1900s until Hardy and Weinberg introduced that idea. That's a very good question. Most bad mutations tend to be recessive, but that doesn't necessarily mean that just because a trait is dominant or recessive that one is derived and one is ancestral. But like, for example, most diseases tend to be recessive. But I wouldn't say, for example, absence of mid-digital hair is a disease, for example. That's probably just fairly random. Good question. Now, the things I was showing you are examples of things that are inherited like single gene traits. But the vast majority of traits out there, the vast majority of them that anybody talks about, for example, eye color, as somebody asked earlier, and hair color, are not single gene traits. But even those are even ones that seem to be inherited like single gene traits. But we know there are many, many genes that affect them. There's lots of contributing genes. Now, some of them may have big effects and some may have small effects. That fictitious example I gave you about height, every gene was having an equal effect. That's not usually true. Usually there will be some genes which have a big contribution, some genes which have a smaller contribution. But very few traits really are truly single gene, aside from a couple of genetic diseases, which we'll come back to that. So, moving on from there, genetics of human diseases. Now, some genetic diseases are not actually caused by genes, but are actually caused by defects in how chromosomes are inherited. So Down syndrome would be an example there, where instead of having 2 copies of one of your chromosomes, you actually have 3 copies of chromosome 21. You can see that in this picture of all the different human chromosomes. That's not going to be our focus here. Now, these are still genetic in some sense, but I'm not going to be talking about these sorts of things. I just wanted to mention that in passing. Now, a lot of other diseases have some sort of genetic attribute to them. So things like sickle cell anemia, which I talked about in one of my previous talks in response to one of the questions. Lactose intolerance, not exactly a disease, but it is something which, again, has a genetic basis to it. Color blindness, I don't know if any of you here, is anybody here colorblind? Yeah, there's a few colorblind folks. So it's hard to read some of the numbers in here if you are colorblind. in those circle links. You guys have probably done those tests before, right? Yeah, that's what I figured. See, it's a design. It's not a number. Yeah, everybody started to worry there. It's in Arabic. No, I'm just kidding. Now, those ones I just showed you are ones that are fairly simple genetic. Other human diseases have a large genetic contribution. So they're not strictly genetic, but there's a large genetic contribution anyway. So, many of you know about heart attack risks. And these are the kinds of things you hear run in families. You'll know that within families, a lot of people may have diabetes, or a lot of people may get various cancers, things like that. Alzheimer's, arthritis, etc. So these are just some examples of things that have a genetic component but are not strictly genetic. Now, the question is, what are these diseases? Now, an important thing, people talk about, and you saw in those headlines, the diabetes gene was found, things like that. No gene functions to cause a disease, right? There's no gene whose function is to do that. So basically, when you see something like that, that when mutated increases your likelihood of having a disease, what's probably happened is there was a mutation that disrupted the normal function of that gene. Right? So in the case of color blindness, you may have something which makes an eye photoreceptor for green not form properly, and that's why you can't distinguish, say, red from green. This is an important facet to it. There's no gene for disease specifically. Now, if the bearer of the mutation lives and reproduces, the mutant form gets passed on. So let me talk about a couple of different modes of inheritance, just applying what we talked about before. So most genetic diseases are not dominant. This one here that's depicted would be a case of a dominant one. So in this case, the father is affected. So the father there has the disease. He's big D, little d. So we're assuming the big D form, the big D allele is what's causing the disease. In this case, half the kids would be affected as well, right? It's just whenever he passes on this gamete to his kids, they get the disease. Mom is unaffected, and none of her alleles would cause the disease, but some of the kids could nonetheless get it. Now, this is the exception. The more common thing is to have something like this, where the father is a carrier. So in this case, the father has the disease mutation. He has the disease allele there, and he passes this on to the kids. But if he has a kid with a mother who doesn't have this variant, then none of the kids actually have the disease. Two of them are carriers, just like dad, but they don't actually have the disease itself. In this case, the disease is recessive. The time when the disease pops up is not when dad is a carrier and mom is not, but instead when both parents are carriers. In that case, again, just like with Mendel's peas, 1/4 of the kids would get big D from dad, so half of his gametes, and big D from mom, half of hers. So this one would be affected. These two would be carriers, and that one would be okay. Okay? So there's several important principles here, and this is very important to what we're going to be talking about later. You can be a carrier for disease without actually exhibiting the disease. This is when you have one copy and not the other, and assuming it's single gene. And you can have kids with disease even if neither parent displays it. This is important when you start pointing fingers at dad. Just kidding. Now, people being carriers for diseases is thought to be why inbreeding is bad and why there's actually been a lot of inbreeding avoidance in humans and in a lot of other species. Because nearly everyone is probably a carrier for some kind of disease. Everybody in this room probably has, like if you were essentially to mate with a clone of yourself, which would never work anyway, if you were to mate with a clone of yourself, there's a high probability that some of your kids would have some disease because you're probably a carrier for at least one disease. The good thing is these tend to be rare. So if you mate with somebody else, there's a good chance they're not a carrier for the same disease that you are, and the kids are all okay. Now, if you mated with a relative, that increases your odds that the kids will have a disease because they're likely to have the same bad variant that you have. And that's thought to be why culturally we've adopted this idea of don't have kids with your sisters and brothers, things like that. It's better to go far out, go to a different place. And in fact, it's been shown in studies that you're about 2% more likely to have kids with a serious disease if you have kids with your cousin. Now, cousin, that's not common in the United States, but in a lot of countries, people do have kids with their cousins. In fact, my grandparents, my maternal grandmother and grandfather were first cousins, and in Egypt there was no avoidance of that. Yes? Basically, you're just more likely to make things homozygous, so the opposite of heterozygous. You're more likely to make a big A, or a little a. If you happen to have something where you have one good copy and one neutral copy, then yes, in that sense, you would be more likely to make good things. But it's much more likely to have the other way. It's possible. It's possible. It's certainly possible you could do that. Yeah. Yes.
Dogs, U.S. survey, veterinary hospital, less than 1 per 100,000 months mixed breeds
Mhm.
have genetic deafness occur.
Huh.
AKC average is approximately 4 per 10,000, and Dalmatians are 30%.
Wow. Well, that's definitely going to be a result of this sort of thing. So the comment here was this: that essentially, when you have breeding within dog lines, you're much more likely to get diseases popping up, and that's the same principle. Yeah, especially relative to a mutt. Yeah. No, that's very good. What this picture, by the way, is at the bottom is this shows years that different states within the United States adopted laws banning inbreeding. You notice the Southeast there never did. But, a lot of the others did, though. So, question is, where do these mutations come from? Where do these bad diseases come from? They're obviously bad. Selection tries to push them out. They typically result from errors in the replication of genes. Now, again, there's 3 billion nucleotides in the human genome. These are the letters I was describing to you before. And every now and then, on order of 2 per 100 million, you have some sort of mutation happening. Now, given that you have 3 billion in your genome, this means there's probably about 60 differences between parents and offspring. 60. That's a lot of mutations out there. Now, the estimates are about 1 or 2 of those detectably affect fitness. So we're continually making kids that have bad mutations that we didn't have. Now, what happens from an evolutionary sense, what happens then is those then get purged by selection over time, but they do happen. And they're, yeah. Those are 1 to 2 bad. The number that are good, it would be much, 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 much lower. And you can think of it, in terms of mutation, you can think of it as the example I always like to use in my classes. Imagine you have a car sitting over there, and I take a hammer and I throw it at the car. There's some probability I will make that car better. But the probability of that is infinitesimally low relative to the probability I'm going to do something pretty bad to it. That's the analogy I usually use in that context. Now, obviously, some treatments will make mutation rates higher, like exposure to UV light or chemical exposure, obviously some sort of radiation. Now, in the popular media, when you talk about mutations, people think of the X-Men and things like from Heroes, things like that. Unfortunately, they're not usually like that. Most mutations are probably far more subtle. That usually what happens, especially for the ones that are causing an effect, there's probably some slight developmental defect. It may not even be noticeable. It may be something just where you're slightly more predisposed to stomach pain. Yes. That's possible. That's possible. I'd have never seen a study of high altitudes to see if they actually show more detrimental mutations or more effects of detrimental mutations, but that's certainly a possibility. I mean, one thing that happens certainly is when you're at high altitudes, you do get more, exactly, you get more blood cells, so which could potentially help you in that context resisting it. Yeah. Now, some of these are going to cause changes in productions of enzymes or hormones, but very often, these are just not going to be mutations that actually show up unless you have multiple risk mutations. This is going to be the factor that's going to be very important for what I'll talk about in just a second. Now, typically, you need many risk mutations together to get these things. Now, aside from cases like sickle cell anemia, I'm talking more about genes like, or diseases like heart disease, diabetes, things like that. There's multiple factors that contribute to them. So the example here is atherosclerosis, which is the narrowing of the arteries with fat deposits there. There's some mutation combinations. Again, combinations of mutations that are associated with high risk, but no mutation individually has been definitively shown to increase your risk. Just when you have this one and that one, things like that. And we'll talk about that more again in just a little bit. So let me touch very, very briefly on this nature versus nurture idea, just because it always comes up. Again, there's no either or debate here. Nobody will say nature, unless we're talking about the magazine. So nature versus nurture is out there. Anything you look at, almost any trait whatsoever, the answer is going to be both. Almost any trait whatsoever. And I know somebody can come up with one that's not, but basically any trait will be both. And this actually leads into the concept of heritability. So heritability is basically the relative contribution of genetics to your trait of interest. So some traits are much more heavily influenced by genetics, right? So things like facial features. That's unlikely to be very much influenced by, what you've eaten growing up. It'll have some influence potentially, but not very much. In contrast, some traits are much more heavily influenced by environment, such as language ability. And there's obviously some like obesity that have strong influences from both. Now, the relative proportion of variation in that trait that is coming from genetics, the relative proportion of variation in that trait that's coming from genetics, that's what's referred to as heritability. So when you hear people talk in the media about a trait having low heritability, that means most of the effect is environmental. Little of it is genetics. If there's high heritability, then there's a high genetic contribution to it. And the thing that makes this even more complicated is that heritability not only varies across traits, as I just mentioned, but it also varies among populations and across time. So you can imagine in a particular population, there may not be a lot of genetic variation for a trait of interest. So for example, among my relatives, the heritability for hair color would be very low because almost everybody has black hair. Right? So there's going to be any change that you have in there is probably not going to be genetic because almost everybody has the same type. So among populations, the heritability can be different. You also have, of course, interactions with the environment, not just strict environmental influences, but having a particular genotype may make you more susceptible to the environment. Right? So you inherit the susceptibility or sensitivity to environmental risks. So an example there would be skin color. As you have darker skin, you're less likely to have UV damage or cancer, okay? On average. Now let me recap a couple of points here before we get into the meat here. First, some diseases are fully inherited, like sickle cell anemia, and that's one that's actually a single gene example. But many others have just some genetic component, and it's usually a lot of genes that are doing this, right? The heritability of these diseases is often low because there are often substantial environmental factors. When I say low, I just mean not 100%. I don't mean very low. And the associations with any genetic component may be indirect. So let's get into the meat of this. What's happened with respect to identifying genetic basis of common human diseases? Coming back to those headlines from the beginning of the talk. Now, the typical way people do this is to use association studies. I'm using this term a little bit broadly for those of you who are familiar with it. The goal is to find the gene variants associated with disease. People typically use one of two designs, or sometimes will combine both. One is to look in families, look in sets of families where, for example, you might look at siblings who have a disease and some other siblings who do not have the disease and try to see what's different between them. Or, more commonly now, population-based association studies, where, for example, I might sample everybody in this room and sequence you all for your whole genome and try to see, among the subset of you that have blue eyes, what is the consistent genetic basis among you all. The idea that you're trying to do here is look for the strength of association between variants at a position in a gene and having the disease or having the trait. That's the principle there. You're looking for the strength of association between a particular variant and having the disease or trait of interest. So the principle behind this is that you don't necessarily have to find the actual gene that's doing this. You can actually just hit close. If you're very close, genes nearby each other on a chromosome tend to be inherited together. They tend not to get separated in the process of meiosis. This is a process referred to as crossing over, for those of you who are familiar with it. So if you see an association between a gene variant and a disease, it may be the causal variant, or it could be something that's just close to the causal change. So this is a good thing where, that like in horseshoes, close sometimes counts when you're trying to localize where a genetic disease or a trait is mapping to. You don't have to be right on top of it. So when you do an association study, this is an example right here where this is a very fictitious example. Let's say you have 7 individuals, and some individuals are sick and some are not. Which of the variants here would be the one that's associated with the disease? Yeah, exactly. The A there, the third one over there. That's essentially what's happening in the course of doing this. These ones, every time you have an A, you're sick, and every time you have a G, you're okay. So this is the idea that is being applied in the case of these association studies. Now, 2 different ways this can be done. One is you might have an idea ahead of time which gene may be involved. So let's say, for example, you're looking at lactose intolerance. You may assume, well, I'm sure lactase is a good candidate gene for this. So you could do that as one example. The other possibility would just be to look at the entire genome. And this is where those direct-to-consumer genetic tests come in, where you essentially look at 1 million letters simultaneously. So this is 1 million out of 30 billion. So it's a pretty small fraction. But the idea is they're spaced out. So even if you're not necessarily hitting a particular disease gene itself, you're hitting close enough to one that you can identify an association. This is sort of the shotgun approach, where you're looking everywhere all at one time. Now, with the family-based designs I mentioned earlier, you compare your affected and your non-affected siblings. So the non-affected ones are the ones who are not showing the disease. The affected have the disease. So here's an example of ALD. You might look at and see that among the affected siblings, 25% had C at one spot, and 15% of the unaffected had it, and say, well, look, there's a difference there in abundance. So maybe that C is either contributing to your predisposition to ALD or is near a variant that is contributing to your predisposition to ALD. This may not look like a very big difference, but this is actually pretty typical, and you'll see that in just a second. With the population studies, it's very similar. So you might have a group of people who have diabetes here on the left and a group of controls on the right. And you just look and see, among the ones who have diabetes, is there a particular genotype that's more common? So in this case, 46% of those who have type 1 diabetes have this HLA-DR variant, whereas among the controls, only 19% do. So people with type 1 diabetes have a 2.5-fold greater likelihood of having this HLA-DR4 type. This HLA-DR4 is just, a particular base at a particular location, like, say, an A at position 322. Okay? So how well has this worked? If you can't read that bottom, it says, scientists have isolated the gene that makes scientists want to isolate genes. That's pretty appropriate there. How well has it worked? Well, many associations have been discovered. This slide's actually a little bit old. I should have updated the number. But there's something online you can look up. It's called OMIMS. That's short for Online Mendelian Inheritance in Man. This actually began, over 50 years ago, where people were identifying heritable disorders and just mapping them crudely within the human genome. This is now searchable online. You can pull this up. It's free. So, it's open access to everybody. You don't have to have an NIH account. And there's more than 12,000 genes on there that were identified as being associated with a disease or a trait or something like that. So if you're interested in looking at it, just Google OMIM NIH, and it'll come right up. Okay, so it seems like things are going well in that context. And there's new associations every month. I just picked the most recent issue of Nature Genetics. It is from September 2011. As you can see there, here's a common variation at this gene influences meningioma risk. Notice these are just like almost adjacent papers within the journal. Analysis of genome-wide association studies of asthma in ethnically diverse North American populations. And then another one, adult asthma in Japanese populations. Something for Graves' disease. Every issue of Nature Genetics is full of these things. And this is just one journal. This isn't even a particularly medical journal. If you look at the medical journals, there's tons more of these. This is just one random journal I picked. So you see these associations coming up all the time. So it seems like we know a lot, but I'm going to question that a little bit. Now, there are a few cases I'll emphasize. There are some cases that have held up extremely well. So, for example, many of you are familiar with the BRCA1 and BRCA2 genes. These are associated with breast cancer risk. Now, if you look at the broader population, something like 12 or 13% of women will develop breast cancer, whereas 50% of women who have an altered BRCA1 or BRCA2 will develop breast cancer. That is one that has shown a very consistent usefulness and is being used all the time for evaluating breast cancer risk, especially in people who have a family history of breast cancer. The vast majority have not done so well, and this, typically they're not been repeatable in later studies, and the effects, are often negligible. They often increase your odds of getting disease by, 1% is a little extreme. I'll say 3% or less. That often the effects are very, very, very small in these cases. The other question to ask is, how useful is this? So let's say, for example, in actually, I'll come back to the usefulness in 1 second. Let me just show you this first. So I said 12% of women will get breast cancer. This FGFR2 gene is known to associated with breast cancer risk. When people report it, they say, oh, look, the heterozygote has a 20% higher risk. The homozygote has 60% higher risk. Those numbers sound very dramatic, right? But when you translate them, what does that mean? You're going from 12% to 15%, or from 15% to 19%. They're very small increases in overall risk. What is really the difference between having a 12% risk for something versus a 15% risk for something? It doesn't really change what you're doing. The other question is, how useful is it? And imagine the year is 2021. Okay, so we're going way in the future. And imagine you're, say, a 22-year-old girl, or lady, excuse me, just finished college. You're sitting over here at the casino. You meet some guy and you're really interested in him, right? You pull out your iPhone 10, right? He leaves the room for a minute. You go, boop. And it pulls out his genotype off his glass. You say, uh-oh. He has the FGFR bad variant there. He has one copy of the little f, right? Whereas you're good. You have both copies of the big F. What does that do to increase the probability your daughters would get it? Well, let's do this. So you are the big F, big F. He is big F, little f. What fraction of your kids would get his little f? Half of them would get it, right? Right? Here's the 4 types you get. So half of them would get the bad allele. Now again, these are the risks of breast cancer. So on average, among your kids, instead of having a 12% risk, which is what would have happened if you had kids with somebody who didn't have this allele, it would be the average of 12 and 15. So it would be 13.5%. See, again, the usefulness of this is so limited in almost any context, in terms of yourself or even in terms of selective breeding, which some people advocate, which I don't advocate at all. But just the advantages here are very, very limited. Yeah. That's true. Yeah, very much so. If those two were boys, you wouldn't even have that. They do get breast cancer, but not as frequently as girls do. That is true. Now the question is, why are these associations so weak? Why is it we're not seeing things like BRCA1 and BRCA2? Well, one possibility is that a lot of these familial diseases are not genetic. That maybe they have something to do with having a common environment. You often have the same environment in your families too. It may be epigenetic, but I'm not going to go there right now. The ones I think are very likely are these ones here, though. That the genetic effects are really small very often. There's many variants within a gene that can cause the same effect, and I'll show you an illustration of that in a second. You very often have interactions between genes, and you very often have interactions with the environment. Now, what can happen, this does happen sometimes, is you just didn't find the right gene. You got unlucky. Statistics worked against you. You said something like the probability of this association was 1 in 1,000, but you probably did more than 1,000 studies, so some of them would just be unlucky. But I don't think that's a main contribution. I think it's more those 4 in the middle. So looking at, for example, this is the one I showed you earlier about the HLA-DR4 gene and probability of diabetes. This 2.5 times greater likelihood sounds really good until you think about, well, how many people get type 1 diabetes? Well, 0.2% of the population has it, so having this changes that to 0.5%. It's not very much, and especially when you think in the context of your life more broadly, your odds of dying in a car accident are 1.2%. Is it really helpful to know that? There's a decent chance it's really not. Often you have multiple rare variants within a gene that can individually cause a disease, and this throws off some studies. So let's say, for example, you're trying to look and see if any of these was associated with disease. Now, what I did in this fictional example, either this or that causes you to have a disease, and that's very common in a lot of cases. But if you were to do an overall association study, neither one would show up that much, because in this case, this one has the A variant and doesn't have the disease. This one, did I say that backwards? Oh, yeah, I'm sorry. This one has disease but does not have G. This one has disease but does not have C. So in those cases, since individual variants can cause the disease and not necessarily everybody has the same variant, this is true actually for BRCA1 and BRCA2. It's hard to pick them up unless the disease is very common. The other thing, now this is a case where multiple variants within a gene can cause the disease. You can also have multiple variants needed together to get the risk. So the one I showed you earlier was atherosclerosis. You need variants of both of these 2 genes. I'm not going to read them. This one and that one together to have the higher risk for atherosclerosis. Neither by itself gives you an increased risk. And this is the kind of thing that people, when they first do the study, might pick it up because, let's say, for example, this was very abundant in the study, but somebody else does a subsequent study and this particular variant wasn't that abundant. You may miss that association. So things just get really complicated out there. There's a lot of heritable components to diseases that have been identified. Some have held up very well, but not very many. The BRCA1 and BRCA2 are really exceptional. In most cases, you have these fleetingly small effects where they increase disease risk by 1% or sometimes up to 3%. So what about these direct-to-consumer genetic tests? That was basically the point I wanted to get around to from this introduction. It's easy to get a lot of data out there. There are a lot of companies who will give you this for under $200, where you'll just take a Q-tip, you swish it around in your mouth, put it in a Ziploc bag, and mail it off to the company. And you get this, this DVD or an online link that gives you tons and tons of information, right? Now, they'll send you the actual genotype, so you'll find out about 500,000 to 2 million spots in your genome, and they'll tell you which of them have been shown to be associated with disease predisposition. These are just 5 of the companies on here, but there are several others as well. Actually, the one I'll be focusing on the most here is 23andMe. I'll pick on them a little bit. This, by the way, is very cheap. When I gave this talk about 2.5 years ago, the price on here was about $1,000. Now it's well under $200, and sometimes I think Julie just had hers done for $60. I think, right, Julie? Yeah, $60. Charge to your room, yeah, that's right. Who knows, it might be a cruise option at some point in time. So the question is, what can you do when you get this information? Right? So the idea that they'll advocate is you can have some sort of personalized medicine. You can begin treatments and monitoring early. You'll know some medicines are more likely to work for you. There's a burgeoning field called pharmacogenomics. This is very exciting, where they'll identify particular diseases that work particularly well. I'm sorry, not diseases. Medicines, I said that wrong. Medicines that work particularly well if you have a given genotype, or medicines that may not be effective or potentially even detrimental if you have a different genotype. And the other thing that can sometimes happen is you can have preemptive surgical removals, and I'll talk about that in just 1 second. The other thing you can do is try to be proactive environmentally. So if you know you're predisposed to diabetes, maybe you might watch your sugar intake, take extra care to exercise, etc. In terms of the preemptive surgeries, there's a 2008 study that showed if you remove your ovaries, it'll actually reduce cancer risk in women with a BRCA2 mutation by 72%, or breast removal by over 90%. So this sounds like it's the kind of thing that maybe if you knew you had the risk factor, it might be worth doing. The question is, this is a really scary prospect, and even the knowledge of this is very scary. In fact, there was a study which showed of 275 patients, and notice who these are, from families known to carry BRCA mutations. Only less than half, only 48%, were willing to undergo genetic testing. They didn't want to know. Like, I don't want to hear it. I don't want a death sentence. You can understand why that would be true, right? You don't want to live your life thinking, oh my gosh, I have a high likelihood of getting this disease. I don't want to have my ovaries removed or breasts removed, things like that. A lot of people don't want to do that, and it's understandable. There's also no guarantees. You can take drastic actions and still get the disease. You might have your breast removed and still get it, right? Something like that could happen. You can also take no action and never get the disease, because even when you have the BRCA2 mutation or BRCA1 mutation, it's not 100% likely you're going to get the disease. It just goes from 12% to 50%. So I call this the expensive airbag effect. So airbags are known to be not particularly effective, but imagine you have to pay a huge amount of money to get one too, where it's not going to be that effective, you may never use it, and even if you do use it, it may not save your life. It's kind of a scary prospect. The other thing is that most of the results won't tell you a lot because, again, most of the effect sizes are very small. They're usually less than 3% and often less than 1%. And some may even be wrong. Sometimes you may even be wrong in that the cause mutation is not the one survey. So let me show you some actual data. So these are data from a study that was done of this. This is actually, I'll show you part of my genotype. So one trait is asthma. I do actually have asthma, so this was kind of a silly one. The general prevalence in U.S. adults, they tend to control it by U.S. I apologize for those of you who are not from the U.S. The general prevalence is 5 to 7%. This is one variant it's associated with. This is just its full name. It's found on chromosome 17, the Q arm. The risk allele is T, and my genotype was TT. So I do actually have asthma, so that wasn't a big shock. Now, when you look at this, that sounds, oh, that could be potentially useful to know when you have a little kid, for example, right? However, this risk increases 45%. So it goes from 5 to 7% risk to 7 to 10% risk. So knowing that doesn't really change your odds all that much, right? I mean, if you didn't, let's say, for example, I didn't have asthma, my odds of developing would move from this to that. It's not very big. The other thing is that 23% of Europeans are TT. So there's a lot of people out there that have that. So let me show you another one. This one, I like this one a lot. This is for high cholesterol. There are 3 variants associated with it. I didn't put the names in there. 3 variants associated with them. There's risk variant marker 1 is C. C is a good one that actually reduces cholesterol. So I have A over C, which is good. At risk variant 2, I have A, which increases cholesterol. Or I'm sorry, A is the one which increases cholesterol. I have CC, so that's neutral. At risk variant 3, C increases cholesterol, and I have CC, which is bad. So what's the takeaway? Exactly. There's no takeaway there at all. And this will happen for a lot of cases because there's a lot of... Exactly. If they're all bad, maybe my odds would go up. But for a lot of diseases that you'll get, this is going to be the kind of distribution that you'll see. So this shows you what the actual data looks like when you log into 23andMe with your data. This is actually altered data. This isn't mine. Mine wasn't actually through 23andMe. But they'll tell you, for example, for particular diseases like age-related macular degeneration. Right? They'll say your risk based on a particular SNP is 11.3%. The average risk in the population is 7%. You know, restless leg syndrome, your risk is 5.2%. The average risk is 4.2%. So this shows you a sample set right there. And if you look at this, a lot of these are, you know, yes, it's higher than average, but It's not particularly useful. Like the stomach cancer one here is from 0.07% to 0.08%. Yeah. So it's very tricky to know how to use this. They'll give you a little bit more detail too for each one. So this is zooming in, for example, on that age-related macular degeneration example. So it shows you like what fraction of people in the population have this. There's your odds calculator. There's the 3 genotypes there on the bottom. Just shows you the 3 possibilities and the average risk. So whether you have increased risk or decreased risk based on your genotype. Now the thing that these are actually extremely useful for is not so much the disease part, but when you look at ancestry. So in this particular example, this is not me, obviously, because this person is fully of European ancestry, no African or Asian. This part, a tiny dot of Africa. It's clearly not me, though. What it will do is it'll give you some idea of your ancestry, and they'll paint different segments of your genome and say, this has an Asian origin, this has an African origin. Some of them, this one's a pretty crude one. Some of them will actually give you a very specific one. So the one I got said, you're very likely of Middle Eastern origin somewhere near Palestine, which my ancestry is from Egypt. So that was pretty good. These actually work extremely well. So if you're interested in genealogy, I actually do recommend this, unlike the disease part. I think they actually work very, very well for that. And they'll tell you if you have mixed genealogies from different types. There, it's actually been very, very good. Unfortunately, you may remember that slide I showed you in the very beginning about interest level. Most people who do it either do it for curiosity, which is fine, or medical risk. Very few did it because of interest in genealogy or ancestry. So even though that's the best reason to do it, is through that sort of thing. Also, if you're truly geeky like me, there's a Firefox plugin such that when you read a paper and it mentions one of these variants, it'll tell you immediately as you're reading the paper, your genotype is TT. So if you're truly geeky, that's a good one. Yeah? Is there a question?
National Geographic study, you find that useful?
I don't know which one you're from. Oh, that yes. That is definitely useful. I agree completely on that. Let me come back to that at the end. Let me just flip through this. Oh, was there a question over there? Sure. It's difficult because, unfortunately, this example is all blue. So what would happen is there would be sections of red if somebody had Asian origin, or sections of green if somebody had African origin. But since this person was fully European, all you see is this big blue. So let me do this right here. If somebody had an Asian, it may look like this, for example, if somebody was Asian for chromosome 8. Yeah. Let me change back to that. There's one little African piece right there. Yeah, I apologize. That example is the only one. I can't get other people's data, so this is what I was limited to. Yeah. There you go. And the other thing is, associated with this, most of the recommendations are obvious. What are they going to tell you? Let's say, for example, you have greater diabetes risk or cancer risk or anything risk. What are they going to tell you? They're going to tell you should exercise. You should maintain a healthy diet with high fiber, lots of vitamins. Watch your weight. Don't smoke. Limit your toxins. Rest. There's no disease that this is not the set of recommendations for, right? There's going to be a couple of specifics out there, but the vast majority, this is going to be the standard recommendations. It's true. I'm not a coffee drinker. That's true. There are some benefits supposedly associated with coffee, and the same with wine. But again, coffee and wine are those mixed blessings, right? Where there's definitely some advantages and some disadvantages. I don't do either one. With wine, I don't care. No, that's right. So the question is, how useful is it to have this knowledge? Is knowledge truly power? Now, thinking about this, what are the risks? So I talked about the benefits, and there's some benefits. I'm not saying there's not, and I'll come back in just a minute about some actual medical benefits. I'll talk about that in just a second. But what are the risks? Well, one is overinterpretation. I mean, many of you here in this room, since you're Scientific American readers, many of you are very good with statistics. You understand when somebody says 2.5-fold more risk, but your odds are 0.08%, that it doesn't mean much. But a lot of people say, oh my gosh, 2.5 times greater risk, and they freak out. Right? And this is the kind of thing that you have to watch for. This is actually what the FDA in particular was very angry about. They said this sort of test should be applied in consultation with a genetic counselor or somebody like that who can calm you down and inform you on it. So people tend to assume this is correct and informative, even if it's not. It'll sometimes create anxiety, or sometimes the opposite, sometimes an undue sense of security. You may think, well, I have a family history of cancer, but I don't have any of the cancer risk, so I'm okay. That's probably not a good thing to do. The other thing is, of course, the danger of misuse of information by others. So one is, personal stigma. I don't think this is very likely, but it could potentially happen. Insurance companies I don't trust at all. I totally would believe they would do something. Or employers not hiring because they say, oh, this person may be likely to get this bad disease. I don't want them. Now, there are protections out there. Oh, I don't know if any of you are familiar with this. Have any of you seen this guy before? Mayhem, yes, good. So this is the kind of thing you might see in an Allstate insurance commercial in the future. I'm a mutation causing rampant, uncontrollable cell growth that can ultimately kill you. This is, for those of you who aren't familiar, this is somebody who shows up in a lot of insurance commercials, and he's always talking about something awful that's happening to you. Usually it's something about like a branch falling on your car and destroying it or something like that. But this is the kind of thing you can imagine in a commercial in the future. But again, there are protections, so at least within the United States, not across the world. But in the United States, there are protections. One of them is the Genetic Information Non-Discrimination Act, also abbreviated GINA. So basically, American insurance companies and health plans are prohibited, this is an important thing, from looking at your genetic information before you enroll, requesting or requiring you to take any sort of test, restricting enrollments, and changing your premiums based on genetic information. This was actually passed almost unanimously in the U.S. Senate, which is extraordinarily unusual. One senator voted against it. Anybody want to guess who? One senator voted against it. Ron Paul. Ron Paul was the only one who voted against this. There you go. Congressman. I'm sorry, I said that wrong. I'm sorry. Congressman. My bad. The other thing is, so that was looking at insurance companies. So American employers, not just insurance companies, are prohibited from discriminating against who they hire, requesting or requiring you to take a test, or disclosing your genetic information. So, and this part's a little bit worrisome right here, though. That's a little bit worrisome in there. The language is a little bit open. Yeah? So I'm going to come to that in 1 second. I think it's the very next slide. This was signed into law in May 2008. But again, this is just the United States, and this is just a subset of the things. Now, the risks are not gone. So one of them, actually, as the gentleman in the back was pointing out, this does not apply, for example, to long-term care insurance. This does not apply to it. It only applies to health insurance directly or employment. So that's there you go. Exactly. That would be the way to do it. As the gentleman in the back said, get the Alzheimer's test after the insurance. But, GINA may go away. I mean, there may be a time where, the Ron Pauls of the country get together and they say, we don't like this. We want to get rid of this law. Consent may not always be fully informed. People say that when they put out these tests that, the person asked to have it, they understood, they consented to having it. They may not fully know what they're getting themselves into. Right? And this is important to that top one there. Basically, how well does the consumer understand what the data is that will be used and shared? Yeah. Yeah. Yes. Yes, they do. But it's anonymized. It's not like, you can find out that this was Mohammed Noor's data. It's just, they do this more for basic research, and it's just, anonymized in terms of people. Oh, yeah, that's right. You do have to agree to participate as well. So the question is, should you try to discover your genotype? This is sort of the ultimate question this entire talk is leading to. Should you try to do it? So this is very much a personal decision. I think there are times when it is helpful. Right? There are times when it's helpful. So one of them would be, for example, if you do have a family history of something like breast cancer, and let's say, for example, that your relatives who have it take it the same time as you do. You may find that your relatives who have it have a BRCA2 mutation that you do not have. That would be potentially useful. Right? Another reason to do it would be in the case of, for example, ancestry. If you want to really understand where your ancestry is from, it works very well in those sorts of contexts. I would definitely advocate it in there. Generally speaking, though, if you don't have any particular risk factors, if you don't have particular family histories for things, I think it's very unlikely to affect your long-term health with the information we have today. This last qualifier is very important. As we start to understand the complications more, maybe 10 years from now, when we start to understand better how these different variants interact with each other, when we try to understand better what the interactions with the environment are, the benefits of these may be quite a bit greater. You may be able to have a much more precise estimate of what your odds are of getting a disease. Right now, I think it's mostly useful for curiosity or interest, or useful for the community if you allow people to use your data. That maybe that in contributing your data to this overall thing, we can get a better handle on some either diseases or even just common genetic traits, things like that. Yeah?
So if the existing
Yes.
they will look at something like 2 million,
Yep.
2 million basements.
Exactly.
And if research identifies interactions or
Mhm.
better associations or something like that, it may not be on the 2 million that they test today.
It's possible. The advantage there is that since you're doing 2 million, you're likely going to have a variant that's close to the other variant. So you may not have an exact risk, but it'll tell you that you're likely to get a risk, because this particular base is 100 bases away from the one that's actually causal.
They do genotype more sites than they actually have associations for right now.
Yeah.
So if an association is found for one of these other sites, they update you, at least with 23andMe, as long as you continued your subscription, they update you.
My question is, if the associations will be stronger in the future,
Mhm.
based upon further research,
Mhm.
is it better to wait until that has actually
Good question. Good question.
or do it?
It depends on the type. If it's the sort of SNP things like 23andMe, I'd say probably in that case better to go ahead and wait. If it's full genome sequencing, which some companies are actually doing now, I mean, the only advantage to waiting is it'll probably be cheaper later. But on the other hand, at that point, there's no bases that are being missed. If it's a full genome sequence, you're going to get just about everything there. So in that sense, it's not much more. It is more, but it's not crazy. I think it's like 1,000 or something like that. I forget the exact price. Or at least that's the intended goal. I don't think it's actually at 1,000 yet, but the goal is to try to get it to around 1,000. It's getting there. It's getting there. So again, there are times when this will work. Now, one important thing I should emphasize too: if you know you have a family history for a disease, that information is far more useful than anything that 23andMe or any of these companies will give you. That is, really, really useful. 23andMe will just give you these essentially these sort of guesstimates. Yeah.
Your picture the other day showing 8 countries that believe in evolution.
Yes.
Iceland was number 1.
Yes.
That's understandable because the whole country is a genetic study. Are we finding anything out from Iceland?
A lot. That's a very good question. The question is, I'm not sure about the connection with the evolution part there, but the question was specifically, are we finding a lot from the Icelandic, where we have a lot of inheritance information, we have a lot of genetic tests done. A ton of studies are using the Icelandic ones. So a lot of the associations with particular diseases or associations with particular traits are coming from the Icelandic populations. The danger there, and this came back to something I said earlier, the danger there is we're interpreting this in the context of Northern Europeans, that variants found in Arabs or in Africans, things like that, may not have the same sorts of effects. So there's always a little bit of a worry there. Yeah.
Would it be possible, not today, but in the future, to get a full genomic makeup done of artificially recreating that DNA, including it?
Potentially. Now, it wouldn't recreate you because, of course, you're the sum of your experiences as well. Yeah. Yeah, in principle, you theoretically could be able to come up with something that would be equivalent I wouldn't even be equivalent, it'd be similar to a twin, because if there's any sort of maternal effects from your mother that may not be genetic, you would be losing those as well. So it'd be something that would be very close to a twin. It would be, in principle, possible to come to something similar to that. Do you have a question, Julie?
I just wanted to point out that everybody's had their papers, and so you might.
Oh, yeah, that's right. Let's come back to the papers in just a second, because I'm almost done with this entirely, so we'll come back to that in one moment. So, this is the set of things we've gone over. The basics of genetic inheritance, genetics of human diseases, nature versus nurture, identifying the genetic basis of diseases, and the benefits of knowing your genotype. Now, I just wanted to emphasize there's a lot of ways to die. This shows the probabilities of death and different things in there. Notice that one of the biggest ones there is heart disease. You got cancer, stroke, etc. But there's a lot of things you can control very well, like motor vehicle accidents, etc. Some of these are disease-related and preventable. Some of them are preventable, but they're not disease-related. And knowing your genotype, I think, may help in the future. And sometimes, but not very often today. Sometimes, but not very often today. So let's go ahead and, how many people, have you already tasted the 2 pieces of paper? Okay, try right now. Try the one that was, did you identify white versus blue?
They can't swallow.
Okay, try them separately, not at the same time, and see if you can taste the difference between the two. That's the key. Don't swallow it. It won't hurt you, but it'll be kind of weird. I know, isn't that crazy? So how many people can taste the difference? Raise your hands. So all of you are PTC tasters. That looks like about 70%. How many of you cannot taste the difference? Looks like it's probably about 40%. So it's pretty close. This is pretty close to the whole overall sample. There was one of them. If you can taste the distance, yeah, if you're a taster, especially there's two kinds. If you're heterozygous, you can sometimes barely taste the difference. So that's what I am. I'm a heterozygote. I can barely taste the difference. But if you're a taster, you know there was a taste. It's like whoa. Yeah, people tend to think it's very strong. So for those of you who are actually related here in the room, did you see any interesting patterns either associated with that or any of the other things I mentioned? Yeah, so you're the same. How about you guys? Were you guys the same? Were you guys the same? He could taste and you could not. Okay, so there you go. So that's something from mom there in your case. Yeah. Yeah? Uh-huh. Ah, there you go. So a difference from further back up. Yes, question there. Oh, absolutely. I think all of them are actually building useful data sets. Yeah. I won't like you said, I won't advocate for a particular one though. Yeah. I'll come back to you. Oh, okay. So that could happen pretty easily in the case of something like this. If you're a taster and she's heterozygous, let's say big H is taste, right? If your parents were both heterozygous, this would make you a taster, and this would make her one of these, the moderate one. Right? So this would be your sister, and this would be you in this case. Good question. Yeah. Oh, I'm sorry, I missed you. I promised you before. Yes. So Rh factor is determined by gene, and so both the A, B, and O, and your plus or minus. It's okay. So your husband positive is dominant. So your husband could be plus minus or could be plus plus. If you're a minus, then you would be minus. So this would be, if you were to look at your kids, for example. I'm not sure if you have kids. Your daughter is actually if your daughter's minus, then we know what your husband was. He was a heterozygote then for sure. So this would be the case where this would be your husband and this would be you. And you should know that because of having children. My daughter. Oh, that's true. Oh no, yeah, that's one of the dangers for that. Yeah. Yes. I have no idea whatsoever. The question is, why is there, or has there actually even been? It's not clear there has been selection on that. That's oh yeah. Oh yeah. Oh, is that right? Oh, there you go. That's strong selection recently. They were a taster for that. Yes.
You mentioned that knowing your family history is much, much more important than knowing your genetic fingerprint.
Uh-huh.
If someone was adopted, how strongly would that change?
So if somebody's adopted, then there potentially is a benefit to actually doing these sorts of tests because, any information is better than no information in that case. But
smaller, is it something going to be maybe a lot more relevant?
I think in the future it'll be more helpful than it is right now. But again, there are a couple of cases. Let's say, for example, you adopted a girl, and it turns out she has one of the BRCA2 mutations. That would be something where there might be some reason for knowing that. But again, in that sort of situation, it'd be very important to consult directly with a genetic counselor and things like that before actually doing it. Good question. Yeah. Ashkenazi Jews, exactly. I mean, it's going to be very different for different traits. So the questions about the Ashkenazi Jews, which have had a lot of genetic tests done on for various diseases. Certainly, there's been a lot of very clear genetic contributions from that. I'm sure there's some environmental ones as well. And again, a lot of these traits, there are very few of them that are purely one or purely the other. Most things you look at, aside from things like sickle cell anemia and things like that, are multifactorial. Exactly. And not just multiple genes, but also genes and environment. So it's hard to identify one versus the other. I mean, I'd be making up a number. I mean, if I was to guess, I'd say 50-50, but I'm making up the number at that point. Let me come back to you, Peter, because somebody had a question over there on the side for a while. Wasn't there? I thought I saw a hand up there. Yeah. Oh, okay. Great. Did I answer okay? Okay. Yeah, go ahead.
That's kind of related to the gentleman
would have the disease. Yeah, yeah.
And with sickle cell anemia, they identified malarial resistance as
Yep.
a beneficial predicting carrier. Is there any work? And I think I saw some headline, and again, like with the
Yeah.
about correlation to this sort of academic intelligence maybe being more.
Yeah, I remember reading that too. I'm not sure how. I haven't read the original paper, so I worry a little bit to comment.
In there maybe as well.
It's possible. I haven't read the original paper, so I feel a little hesitant to actually comment directly on how good a study that was or anything like that. But I know the one you're talking about for sure. It's a good question. Yeah.
When we send back the profile papers from these companies, do they take into account your age? Because there's such a long lead time with a lot of these diseases.
Absolutely.
Your age bring difference.
Absolutely. Absolutely, yes. Absolutely, yes. That's a very good point. So the question, for those of you who didn't hear, was whether they take into account age on these things. They absolutely do. Good question. Yeah.
All right, so the mechanics of this, you get one chromosome from mom, one from dad.
Yep.
Each one codes for a different set of proteins.
Well, the chromosomes code for the same proteins, but you just get,
Right.
yeah,
But some can do
Yeah.
proteins, some cannot.
potentially, yes.
And if one of your chromosomes can make a protein, you can make that protein. Is that true?
That's typically true. There are a couple of sort of funky exceptions, but typically speaking, that is true.
It's also true of the XX chromosome.
That's the kind of funky exception that females have X in activation, so only one X is actually producing. But it's like calico cats. It's not going to be the same one in every cell of your body. Yeah, yeah.
Is there a company that's particularly good with ancestry if that's all you're interested in?
I say I'm hesitant, at least in a public forum, to give a specific recommendation for a company. But if anybody wants to talk one-on-one, I'm happy to do more of that. Yeah. They're pretty good. It depends on the company. Some of them give you very, very precise ones. Some of them just give you Europe, Asia, and Africa, things like that. They're pretty good, though, in terms of the accuracy. In cases where it's been known, it's pinpointed pretty good. Yeah. It's ignored. So the question is, how does epigenetics feature into this? It's ignored in this. Yeah, it's ignored in this. They don't usually test for that. Yeah. I have to work out the cross on that to see. I mean, if it's just father-sister, I would imagine there's probably some sort of sex-limited expression that the father is actually passing it on, but because he is male, he's not expressing the particular trait, and then therefore he's passing this on, this trait that his sisters also have, but it's sex-limited in expression. It only shows up in females. But I have to do the actual crosses to see.
The mother's brothers for sons would be more.
That would be more for that. Exactly. Are we okay on time? Oh, sure. Okay. Last question. Or not? Oh, yeah, sorry. I think it's yours. Is it? Mm-hmm. Hmm. Mm. Mm. I love this. Sure.
I don't know if that works because I can't taste it and I hate those vegetables.
So the comment was made about the potential value for the being able to taste these things. Is a lot of those tastes show up in some of the mustards that people have strong reactions to, like broccoli and Brussels sprouts and things like that. So maybe there was some advantage to it could go the other way. It could go either way. No, exactly. There doesn't have to be advantage at all. It could just be truly neutral and segregating it there. So I put this picture on here just because I thought it was pretty funny, but this shows you like a futuristic idea of coming off the shelf and picking your beauty chromosome or your IQ chromosome, things like that. Thank you all for your attention. I appreciate it. If there are more questions, too, I'm happy to talk with you all one-on-one in the lab tomorrow for any of you who come for that.